Okay, now let's talk about section 2 of chapter 10, more specifically about the process of cell division. Um, so we need to talk a little bit about chromosomes first because when cells divide, the cells need to deal with the chromosomes. Um, so chromosome is a structure that contains DNA and protein. The DNA is essentially packaged with the protein to comprise the chromosome. Bacteria, that is prokaryotes, contain a single chromosome, a single circular chromosome, um, whereas eukaryotes contain multiple linear chromosomes. Ours are not circular. In eukaryotes in particular, the chromosomes are packaged with proteins called histones, and you can see, and we're going to study the structure of the details of DNA more later, but you can see the DNA is essentially wrapped around this cluster of these proteins, and that little cluster of proteins with the DNA wrapped around it is called a nucleosome. And what happens is these nucleosomes cluster together in these coils here, and then the coils sort of wrap up into what your book is calling a supercoil. And when we see chromosomes, that is when we stain a cell and can see the chromosomes, we're seeing basically the DNA and the histone proteins tightly packed together. And I want you to take note here that what we're showing, and this is probably kind of structure that you often think of when you think of chromosomes as looking like that. Well, what you're actually seeing is a duplicated chromosome. And that is that chromosome consists of two separate strands, or what are called sister chromatids. So here's a chromatid, here's a chromatid. And together, two chromatids make up a duplicated chromosome, and we call them sister chromatids because each of those chromatids is an exact copy of the other. So when we look at a chromosome that looks like this, we're seeing it in a state at a particular stage of cell division right before the cell is going to divide, okay? And right before those two sister chromatids are going to be pulled apart. Okay, so keep that in mind. That's a duplicated chromosome. A chromosome can just as easily be a single strand. In fact, the majority of the time it is. It's only right before cell division that we produce these duplicated chromosomes. All right, so the cell cycle. This is the cycle a cell goes through from when it's made to when it grows and gets ready to divide and divides again. In bacteria, of course, one cell produces another, and when that new cell has been produced, it will grow. And in preparation for division, it will duplicate its DNA or duplicate its chromosome, essentially. And when prokaryotes divide, they go through what's called binary fission. They duplicate their chromosome. They have the two copies now, the two circular chromosomes, and the cell elongates, pinches off in the middle, and produces two new cells. This is a relatively simple process compared to what eukaryotes do. Eukaryotes do not do binary fission. Prokaryotes / bacteria do binary fission. All right, so let's look at the cell cycle in eukaryotes. So here's sort of the overall look at it. If we start here, and we head this way, we see that after a cell is produced, of course, it has to grow. And so this first phase of growth is known as the G1 phase. The G actually stands for gap, but we can think of it its growth. Um, Sometimes a cell will just stay in this phase if it's not going to divide again, and it just does whatever it does that type of cell does, whether it's a kidney cell or a liver cell or whatever. Um, Now, if that cell is going to divide again, it enters the next step of what's called interphase. And you notice this G1S and G2 phase are known as interphase. That basically comes from the fact that it's the phase, these are the phases in between cell divisions. Okay? So after G1, you have the S phase, and that's when we replicate the DNA. We make copies of each chromosome. So during the G1 phase, each chromosome would just be one linear strand. But after the S phase, after we duplicate them, each of those chromosomes will consist of two strands or two chromatids and will be duplicated chromosomes. Okay, so after we duplicate the DNA, those chromosomes, we enter the G2 phase. This is where the cell is preparing for mitosis. It will, if necessary, produce more organelle. It can duplicate things like mitochondria, for example, in preparation for that cell getting ready to divide. All right, so then after G1, S, and G2, this interphase, we then enter official cell division. And we're going to do mitosis next. Mitosis has a series of steps, and that's why this cell division phase is known as the M phase, because it's essentially sort of dominated by mitosis. And mitosis is not officially the splitting of the cell, but you can think of it more officially as the division of the nucleus. Okay, in preparation for the cell to split in two. Cytokinesis is the official process where the cell splits into two. All right, so let's look at the steps of mitosis. And so in the cell cycle, we have, again, interphase with G1, S, and G2. And during mitosis, we have several phases, and we can think of them as PMAT, prophase, metaphase, anaphase, telophase. And then at the very end of the cell cycle, we have cytokinesis, which is the splitting of the cell. And again, mitosis is the splitting of the nucleus. All right, so let's look at mitosis. So in prophase, what's happening is the nucleus, the membrane, the nuclear envelope, if you will, begins to break down and essentially gets out of the way because we don't want it there when we're going to split in two. The centrioles, specifically these are in animal cells, they're structures that make microtubules. And the green things, what collectively are known as is known as the spindle, is a bunch of microtubules that are going to be used to manipulate the chromosomes. During prophase, the chromosomes also condense. During interphase, if you stain a cell, essentially the nucleus, that is, you stain the DNA specifically, the nucleus just shows up all dark because the DNA is spread throughout the chromosome. It's in a very diffuse state, what is known as chromatin. Now, the chromosomes are there, it's just that they're really long and thin and just kind of spread out and wrapped around each other. And so when you stain the nucleus, you just kind of see this big mass of DNA. And at that state, again, we refer to the DNA, the chromosomes, as just chromatin. Now, during prophase, the chromosomes condense, and you can now begin to see the individual chromosomes. So again, now, of course, our nucleus is beginning to break apart, but now we'll be able to see those individual chromosomes, okay, because they've condensed. And at that point, we don't refer to it really as chromatin anymore. Centriole has split into two, and one is moving to one side of the cell, one is migrating to the other. So that's what happens in prophase. The nuclear envelope breaks down, chromosomes condense, you can now see them. Centrioles produce the spindle fibers or the microtubules, and they begin to move to opposite ends of the chromosome. In a duplicated chromosome, the part where they're attached is the centromere. That's what we're pointing at there. All right, next, metaphase. So now the nucleus envelope is completely broken down. What happens is a microtubule attaches to each side of one of these duplicated chromosomes at the centromere. Okay? And the chromosomes, the duplicated chromosomes, are lined up along the center of the cell. Okay, so that's metaphase, the state at which they're sort of lined up with all the microtubules or spindle fibers attached to each chromosome, one coming from each end of the cell. Now here we're just showing 4 chromosomes, but of course in us there would be 46. We're just keeping it simple here. Anaphase. Now what's happening is notice that the duplicated chromosomes, here's this big one here, has been pulled apart. That is, those sister chromatids have been separated and are now moving or pulling, being pulled, if you will, to opposite ends of the cell. So each of those duplicated chromosomes is now separated. Now, at this point, we really don't refer to them as sister chromatids anymore, but they are distinct, separate individual chromosomes. So whereas up here we have 4 chromosomes, officially we now have 8 chromosomes. They're each consisting of a single strand. They're not duplicated chromosomes anymore. And with this hypothetical situation here, we do have a cell that essentially has 4 chromosomes. All right, telophase. So we're at the end of mitosis. So now what happens is those chromosomes have been pulled to opposite ends, and the nucleus begins to reform. Okay, so now we have this brief moment in time where this single cell has essentially 2 nuclei. But what happens? Now the cell begins to pinch off, and it's the cytoskeleton that's involved with helping to manipulate that membrane. Remember the cytoskeleton when we talked about cell structure? So the cytoskeleton begins to sort of split the cell in two, and that's what is cytokinesis. When in the center it pinches off and we get the membrane, what was at one point the membrane that was like that, as it began to pinch off, at the end of cytokinesis, now you essentially have 2 separate unique cells that will have essentially the same number of chromosomes, the same DNA, because the DNA has been copied. And so it's at this point where we will have clones, 2 cells that are identical to each other. Now in plants, there's a little bit of a difference in that, of course, you have a cell wall. And what happens is after the cell membrane pinches off through cytokinesis and you have 2 separate cells, you have to rebuild a cell wall between them, and that's what we call the cell plate. That is, it essentially the cell wall now builds between those 2 separate cells. So that's something you have in plants that you don't have in animals, because of course animals don't have a cell wall. All right, so I'm running out of time on this video, but here's the summary. You want to take a look at this with all these stages. Interphase, where the cell is growing, duplicating the DNA, getting ready for division. Prophase, where we get started with the chromosomes condensing, the nuclear envelope beginning to break down, those centrioles forming and moving to opposite ends. Metaphase, the spindle fibers have moved the chromosomes to the center. In anaphase, we pull them apart. Telophase, we form the 2 new nuclei. And cytokinesis, we split in 2.